Very good morning to all. Myself, I am Pradeep, lecturer in Biotechnology, and welcome to Government Degree College Men's Srikakulam YouTube channel live. Today we are going to discussing about polymerase chain reaction and its types and applications. These are the topics we are going to discussing under PCR, that is brief introduction about DNA, primer characteristics and its design, basic principles in PCR reactions, types of PCR and applications of PCR. First of all, we will know about DNA. What is DNA? You all know DNA is the deoxyribonucleic acid, but the exact name of DNA is D beta 2' deoxyribonucleic acid. This is the IUPAC name for the DNA. What is the full form of DNA? DNA is the D beta 2 prime deoxyribonucleic acid. What is this mean? First of all, we will know about D. What is the meaning of D? If you take DNA, it contains polynucleotide strands, 2 polynucleotide strands, and each polynucleotide strand contains nucleotides. Nucleotides contain sugar, that is ribose. This is the ribose sugar which contains 5 carbons, and because of the 5 carbons it is called ribose sugar. And this is first carbon, this is second carbon, third carbon, fourth carbon, and fifth carbon. Among these 5 carbons, the fourth carbon, which is immediate to the last carbon, is known as penultimate carbon. Which is immediate to the last carbon is known, fifth carbon is known as penultimate carbon. That is, the fourth carbon is known as penultimate carbon. In this penultimate carbon, if the OH is present on right side, then it is called D configuration of ribose. If the OH present is on left side, then it is called L configuration. In DNA, the ribose is in T configuration. That's why this is called D. Because why we are considering D? Because here the ribose present in the DNA, the penultimate carbon contain OH group on the right side. In the ribose sugar present, which is present in the DNA, in the penultimate position, that is fourth position, the OH group present on the right side. This is known as D. That's why we are included the name D in the ribose sugar. Next, what is the meaning of beta? Here we take, this is ribose sugar, same as before, and in the first position, the carbon contain aldehyde, that is C double bond group, and the double bond present between carbon and oxygen. The carbon has electropositivity, whereas oxygen has electronegativity, which is high electronegativity. Due to its high electronegativity, it attracts the electrons present in the bond and gets partial negative charge, and the carbon gets partial positive charge, like this. Because of the attraction of electrons from the bonds towards oxygen, carbon gets positive charge and oxygen gets negative charge. Due to fulfillment of valency of oxygen minus, the penultimate OH group containing hydrogen transfer to oxygen group and become like this. And in order to fulfill the carbon valency, which is 4, a bond is formed, glycosidic bond formed between the fourth carbon and the first carbon. This is known as glycosidic bond, and this is known as anomeric form. Due to the formation of anomeric form, an extra asymmetric carbon formed. The first carbon initially it was not asymmetric, but due to the formation of a anomeric form, due to the formation of glycosidic bond or migration of electrons towards oxygen and form bond between first and fourth carbon, there is extra asymmetric carbon will be formed. If the asymmetric carbon contain, the first newly formed asymmetric carbon contain OH group in the right side, then it is called alpha. If the OH group present on the left side, then it is called beta. In this, the DNA ribose contain the OH group on the left side. Actually here is in right side, that is alpha. But in DNA, in the case of DNA, the ribose sugar contain the first position OH group on the left side. That's why it is beta. In this way, D beta came in DNA because the ribose sugar, the penultimate carbon contain OH group on the right side, and the newly formed anomeric carbon, the OH group present on the left side. That way it is called beta. And what is the meaning of 2 prime? 2 prime means here the ribose sugar, the anomeric form. Here the ribose sugar, if you take this as a Howarth structure of a ring form of the ribose sugar, this is the Howarth structure of ribose sugar, and there is a glycosidic bond between first and fourth carbon. This is first carbon and fourth carbon. There is a glycosidic bond. This is the glycosidic bond, and this second position which is only contain H but not O. That means it is reduced. So it is called deoxy. In DNA, the ribose, the second position OH group doesn't present but only present H. Oxygen is absent. That's why it is deoxy. Where deoxy condition is there, exists only 2' position. That's why 2' deoxyribonucleic acid. So this is the full form of DNA. That is D beta 2' deoxyribonucleic acid. Why D? The ribose sugar present in the DNA contain the penultimate position OH group on the right side. So it is B. Why beta? The newly formed anomeric carbon contain the OH group on the left side. That's why beta. Why 2'? In the second position, instead of OH, only hydrogen is present. So 2' deoxyribonucleic acid. This is the explanation for the why the name DNA given like this, D beta 2' deoxyribonucleic acid. Next, second one. This is the DNA double helix structure which contain 2 polynucleotide strands linked by hydrogen interactions, and this is the single polynucleotide strand, and these are the nucleotide bases. These 2 polynucleotide strands attached to each other by ionic interactions, hydrogen interactions, van der Waals interactions, and hydrophobic or base stacking interactions, and all these interactions are weak interactions. And each polynucleotide strand contains nucleotides. That is, a polynucleotide is the polymer of nucleotide, and each nucleotide contains a sugar, a nitrogen base, and phosphorus. So 3 components present in the nucleotide. Those are sugar, nitrogen base, and phosphorus. And during polymerization of DNA from nucleotides, there is a bond exists between 2 nucleotides that is known as phosphodiester bond. Each polynucleotide contain number of nucleotides, and each nucleotide linked with adjacent nucleotide with phosphodiester bond. So phosphodiester bond is the main covalent bond that involved in the polymerization of polynucleotide strand from nucleotides. So each the bond present between 2 nucleotides is known as phosphodiester bond. Nucleotide linked to each other by phosphodiester bond. And each nucleotide contains a sugar and nitrogen base and phosphorus, as I said earlier. And sugar and a nitrogen base linked each other by a separate bond known as beta N glycosidic bond. Why beta? As I already told, the sugar present in the DNA is in beta form, and the nitrogen base linked with to the sugar in the N nitrogen. So beta N. Here beta in nitrogen base, nitrogen atom linked to the sugar. So it is known as beta N glycosidic bond. In this, the carbon pentose sugar or ribose sugar, the first carbon involved in the bond in order to bond with nitrogen base, and in nitrogen base there are 2 nitrogen bases present, pyrimidines and purines. Pyrimidines linked with 1 position, that is N1 position. So in case of pyrimidine nucleotides, the bond name is C1 beta N1 glycosidic bond, whereas pyrimidines, the 9th position is linked with the sugar. So in case of purines, C1 beta N9 glycosidic bond. Okay. In case of pyrimidines, first position of the nitrogen base linked with the carbon of the pentose sugar, whereas in case of purines, 9th position linked with the sugar. In this way, the bond involved in this beta N glycosidic bond, and the once sugar and the nitrogen base linked each other is known as nucleoside. Upon binding of phosphate to this nucleoside, it gives nucleotide. If 1 phosphate is attached, that is nucleotide monophosphate. 2 phosphates attached, nucleoside diphosphate, whereas 3 phosphates attached, nucleoside triphosphate. The first phosphate linked to the sugar in 5th position by phosphoester bond. That is 5th position linked by phosphoester bond, whereas 2nd and 3rd phosphates linked to phosphoanhydride bond, which are high energy bonds. In this way, the first the first phosphate attached to the ribose sugar at 5th position by phosphoester bond, whereas second and third phosphates linked to sugar by phosphodianhydride bonds. In this way, there are different types of bonds exist in the DNA. First one is in between nucleotides there is phosphodiester bond, which is covalent bond, and between sugar and the nitrogen bases, that is beta and glycosidic bond. Whereas in case of sugar and phosphate, the first phosphate linked to sugar by 5th position by phosphoester bond, and the second and third phosphates linked by phosphoanhydride bonds. These are the different types of bonds exist in the DNA molecule. These are the purines and these are the pyrimidines. Adenine and guanine present are purines. Guanine, thymine and cytosine are pyrimidine in case of DNA. This is the deoxyribose sugar. Once this sugar, here we see the second position contain only hydrogen. That's why it is 2' deoxyribose. This ribose linked to nitrogen bases first position, whereas for with phosphates by 5th position. And a diploid human cell contains 2 meters length of DNA. Actually, if you take adult human cell which is in diploid condition, it contains 2 meters length DNA. Each individual cell of adult human contains a DNA of length 2 meters. If you take measurements of the cells present in the human, if you take a plant or animal cell, it approximately size of the human cell is 100 microns, but it contains in its nucleus there is a 2 meters length DNA present in the nucleus. How it's possible? We can derive the length by using this. If you take the DNA structure, the DNA contains helix. Each helix contains 10 nitrogen bases approximately, and the helix length is 34 angstrom units or 36 angstrom units approximately, and each nitrogen base length is 3.4 angstrom units. Which is the Watson-Crick model of DNA. So each base pair length is 3.4 angstrom units. The distance occupied by the each nitrogen base 3.4 angstrom units. So that is equal to 0.34 nanometers. 3.4 angstrom units are 0.34 nanometers in case of nanometers. We can write this as 0.34 into 10 to the power of minus 9 meters. In case of nano, we can write 10 to the power of minus 9 meters. That equal to 34 into 10 to the power of minus 11 meters. 34 into, if you take the 2 by 100, then it become 10 to the power of 34 and 10 to the power of minus 11 meters. So 0.34 nanometer, we can write it as 34 into 10 to the power of minus 11 meters, which is each length of a base pair. And human diploid cell contains 46 chromosomes, as you know, 44 autosomes and 2 allosomes. And all these chromosomes contain 6 into 10 to the power of 9 base pairs. The 46 chromosome comprises of 6 into 10 to the power of 9 base pairs. If you want to find the length, we can multiply the length of each base pair into total number of base pairs present in the diploid cell. 34 into 10 to the power of minus 11 meters, which is each base pair length, into 6 into 10 to the power of 9. This is the total number of bases present in the diploid cell. It will give 34 into 10 to the power of minus 2. It is minus 11, it is 9. By cancelling, give minus 2 into 6 meters. And finally, you get 2.04 meters. In this way, each diploid cell nucleus contains 2 meters length, approximately 2 meters length of DNA. But it is repeated foldings during chromatin and chromosome formation. It will adjust it to the 10 microns, 100 microns cells. So in this way, each diploid cell contains that much length of DNA. And this is the cell, and this is the nucleus. Nucleus contains chromosomes, and each chromosomes contain genes. There are several genes present in the DNA. These are the different types of genes present in the DNA. That means they are scattered in the DNA. DNA is like, if you take this, the DNA is like the ocean. and genes like fishes. If you take DNA as a version and the genes present, all the DNA is not contained, all the DNA is not considered as genes. Only few are considered known as genes. Considered as genes, and these can give characters to the organism. So if you take whole DNA as a ocean, the genes considered as like fishes. See this picture. Here the blue color one is ocean, which is DNA. We want to find the gene. The fishes are genes. If you want to find the desired gene or targeted gene, we need to add some bait. What is the bait? Primer. What is the bait? Primer. If you want to find the desired gene from the DNA ocean, we need to add some bait, that is called primer. So primer is a short single stranded DNA sequence. So primer is a short single stranded DNA sequence which is used in PCR technique. But in PCR technique, primer used. What is the primer? Primer is a short single stranded DNA sequence that is used in the PCR technique for selective amplification of gene. As I said earlier, in the DNA stage, there are genes are scattered here and there. If you want to find the selective gene or desired gene from that ample number of genes, we need to add specific primer which is specific to that particular gene. In the PCR method, a pair of primers, one is forward and reverse, as DNA is double helix, double stranded. So we need to add two primers. One is forward primer, another one is reverse primer to hybridize the sample DNA region. Whatever the region or whatever the gene you want to isolate or amplify, that specific primer you need to add during PCR to amplify that specific gene. This is our DNA version. It contains several genes. If you want to amplify specific gene, you need to design primers for that specific gene. Then these primers are, one is forward primer, another one is reverse primer. Once primers attached to that specific sites and amplify the specific gene. This is the PCR. So if you wanted to perform the PCR and amplify or isolates your selected or desired gene, you need to design primers for that specific gene. That is known as considered as baits for that gene. But what are the precautions for designing the primer sets? Primer set means forward and reverse primer you need to design. What are the precautions you need to take before designing while designing the primers? As I said earlier, these are the primers. Ideal amplicon length in PCR is 100 to 1000 base pairs. If you want to amplify the target region of DNA, the ideal length should be 100 to 1000 base pairs. So the ideal length of the target gene or target DNA region should be 100 to 1000 base pairs. What is the amplicon length? The amplicon length is the region of DNA present between the forward and the reverse primers. This is the forward primer and this forward primer and this is reverse primer. The region present between these two primers, forward and reverse primers, is known as amplicon length. The amplicon length should be 10 to 100 base pairs in case of PCR for ideal amplification. And the second thing is, primers should be attached at 3' ends of the target or template DNA strand. Always your primers, either both forward and reverse primer, both primers should be attached at 3' end. Here we take, this is the reverse primer and this is the forward primer. Both primers attached at 3' side. So you need to design the primers like that. Those primers should attach at the 3' ends of each strand. Both the primers should attach in the 3' ends of the respective strands. And the TM value for the primers is between 50 degrees centigrade to 65 degrees centigrade, and the difference should be less than 5 degrees centigrade. What does this mean? The TM value, which is the melting temperature for the primers, which is essential for the attachment of the primers to the DNA template. If you want to attach these primers to the DNA template, desired target DNA template, the TM value should be 50 to 65 degrees centigrade, in between 50 to 65 degrees centigrade, and the difference between the primer's PM value should be less than 5 degrees centigrade. One thing we need to remember here that the both forward and reverse primer need not contain same TM value. For example, this is reverse primer may contain 60 degrees centigrade as a TM value, and the forward primer may contain 65 degrees as a TM value. So there is no need to that both primers should have a similar same TM values. One primer may contain one value and another primer may contain another TM value, but the difference between these two TM values should be less than 5 degrees centigrade. This is the thing we need to remember while designing the primer. And primer length should be 16 to 22 base pairs and need not be same length. The primer same, that is why the forward or reverse primer, the length should be in between 16 to 22 primers, but need not be the same length. Both forward and reverse primer need not be in the same length. For example, this reverse primer whose length is 17 bases, and this forward primer whose length is 20 base pairs. There is no need to be both the primers have the same length. There may be chance they may be in different lengths also, but primer length should be in between 16 to 22 base pairs. Next, primer annealing temperature equal to average of primer TM plus or minus 2. What is primer annealing? While running the PCR, we need to adjust the annealing temperature. Annealing temperature means the temperature that is required for the attachment of the primers to the template stand. This is known as annealing temperature. While running PCR, how much temperature we need to set for annealing purpose, primer annealing purpose or primer attachment purposes. Average of primer TM value. As I said earlier, both the primers need not have the same TM values. So if two primers have different TM value, just take the average of those two values and plus or minus 2. You set the plus or minus 2. For example, if two primers have the average, one is 60 and another one is 62, then the average of 60 and 62 is 61. So if you run the PCR, you need to put 61 or 59 or 63. So the average of the TM values plus or minus 2, either 2 degrees centigrade less or 2 degrees centigrade more, or exact temperature with average temperature we can put for the TM annealing temperature during PCR running. And the GC content of primer should be greater than 50%. Each forward and reverse primer must contain GC content more than 50%. You should remember this thing while designing the primer. And the GC clamps. GC clamps means either G or C nucleotides at least 5 bases of a primer. Last 5 bases of a primer. To design a primer, there are 16 nucleotide length of primer is there. The last 5 nucleotide must contain at least one G or C, but for specific binding. But stretch of 3 G or C's need not be present, should not be present. If they are present, they interfere with the annealing temperature and improper binding may take place. So at least one G or C must be present last 5 in the last 5 bases of the primer. This should be for specific binding. And need to avoid self-complementarity. Self-complementarity means instead of binding the primer to the nucleotide stand, template stand, the primer itself form hairpin loop structure. This is known as complementarity. This should be avoided. The primer should not form hairpin loop like structure. So we need to avoid self-complementarity of that primer. If self-complementarity takes place, the primers cannot bind to the template stand. And also avoid primer dimers. That means the primers, the forward and reverse primers should not bind each other and form dimer. So this is also we need to avoid while designing the primer. So there are several softwares for designing the primers. Those are primer is one software, and qPCR another software, AligoCalc another software, AlleleID is another software, and PrimerPlex. These are the different types of softwares, freely available softwares for designing the primers. This is all about the primers. Primers are designed using AligoCalc. How we can design a primer using AligoCalc? AligoCalc is the before designing the primer, we need to go for NCBI, which is National Center for Biotechnology Information, and it is repository for the all types of DNA sequences, protein sequence, all the things. So we can get, in order to design a primer, you need to know the sequence of that gene. So where that gene sequence is available, you can get that gene in the NCBI. First go to NCBI and click on the PubMed. In PubMed, you type the whatever the gene you want to design a primer. If my interest is human insulin gene, here I type human insulin gene, and this is the sequence. In sequence, we need to get FASTA format. First, it will give gene bank format, but for primer design purpose, you click on the FASTA format, and it will give this is the FASTA format of the human insulin gene, and this is the sequence. This is starting from and it is ending. So in this way, this is the sequence of gene. From this gene, we select a stretch of 16 to 22 nucleotides from starting region, which is considered as the forward primer. Select the 16 to 22 nucleotides from the gene starting area and paste in the AlgoCalc software. In AlgoCalc like this, if you click type the AlgoCalc in Google, it will give this type of window. You first select the 16 to 20 nucleotide bases from this sequence and paste here and click on the calculate. If you paste the sequence and click on the calculate, it will give various parameters like GC content. This is the TM value, and this is the length of the primer. Whatever the parameters, as I said earlier, all the things should appear here. If once we satisfied from that values, next click on the check self complementarity. Then it will give window like this. If after clicking the self complementarity, here we will give three things: potential hairpin formation, complementarity, or potential annealing unit. If you get a none for all these three things, then that primer is fit for PCR, and we can consider it as a forward primer. Then take another sequence which is later to the first sequence and paste in the box and click on the swap stand, which is for reverse primer. For forward primer, you need to click on the calculate, whereas for reverse primer, we need to click on the swap stand. It will give reverse primer, and also can check complementarity, and it will give like this. So if both conditions are satisfied, then we can go for this sequence, these primers for order. Once we order, it will within week or 10 days, they will supply the primers and use it for PCR. This is the way of designing primer. Next, basics in PCR. As I said earlier, PCR is the polymerase chain reaction, and it is an in vitro amplification method. In vitro means we are conducting this in laboratory by using separate instrument. So it is known as in vitro amplification. In vivo conditions, the amplification of DNA occurs by replication, whereas in vitro condition, this is amplified using PCR technique. So it is an in vitro amplification method. The instrument used for this in vitro amplification, that is PCR purpose, is known as thermocycler. So the instrument or apparatus used for the PCR is polymerase chain reaction is thermocycler. This is the thermocycler, and this is used for the screen monitor used for the writing the program for running the PCR, and where this is the portal where we put the sample here. And what are the different types of components we need to add for the PCR? This is DNA template. DNA template means whatever the target DNA you have, that is you need to add. From this target DNA, we need to add specific DNA portion. We need to amplify specific DNA portion. That specific DNA amplification purpose, we need to add specific primers which are attached to that specific portion. These are the primers, set of primers. One is forward primer and one is reverse primer. First DNA template, then primer, then DNA polymerase. In order to amplification purpose, we need to add tag polymerase, DNA polymerase. A special type of polymerase used, that is tag polymerase. In case of our in vivo conditions, general DNA polymerase used for the amplification purpose, but in thermocycler we are using extreme temperature. So our normal DNA polymerase or human DNA polymerase is not tolerate that temperature. So we can isolate the special type of DNA polymerase, tag polymerase, which can sustain over 100 degrees centigrade also. So a special type of DNA polymerase known as tag polymerase used in PCR mission, and these are nucleotides for adding ATGC, DNTPs, deoxyribonucleotide triphosphates. We need to add to the PCR for addition purpose, amplification purpose, and in order to maintain the constant pH and other conditions, buffers and cofactors we need to add. Buffers and cofactors means magnesium generally we need to add. These are the all components we need to run the PCR. And all these components take into one PCR tube. This is PCR tube, and each PCR tube contain target DNA, and this is the target DNA, and this is the template DNA, and this is our target gene, and the primers for the target gene, and these are the nucleotides, DNTPs, and the DNA polymerase and buffers and CCL2, and all these components added in the PCR tube, and put the PCR tube into this place and close the mission and run the program. Polymerase chain reaction is a chain reaction, it's a cyclic process. So polymerase chain reaction is a cyclic process, and each cycle contains 3 steps. Those are denaturation, extension, annealing, and extension. So each polymerase chain reaction contains a cyclic reaction which contains 3 steps. Each cycle contains 3 steps, and a series of cycles occur in PCR. So each step contains 3. Each cycle contains 3 steps. So the denaturation, where the double-stranded DNA become separated and become single-stranded, and annealing, where the primers attach to the target DNA, and extension, where amplification takes place. This is one cycle. This type of cycles occur repeatedly. That's why it is polymerase chain reaction. Polymerase chain reaction. So denaturation, annealing, extension. This is one cycle. Next, again denaturation, annealing, extension. In this way, the series of cycles takes place, and each cycle the target DNA, which is mother DNA, become doubled. First cycle, one DNA molecule become 2, and the second cycle 2 become 4, third cycle 4 become 8. Like this geometric fashion, there will be increasing of the cycle. For amplification purpose, ideal cycles is 30 to 35 cycles we generally put per PCR, which will take 1 hour to 1 to 1.5 hour time. And each cycle generally span for 2 to 3 minutes. And lid temperature should always be 105 degrees. For denaturation purpose, we are generally using 95 to 98 degrees centigrade. For annealing purpose, 50 to 65 degrees centigrade we are using. For extension purpose, 72 degrees centigrade we are using. These are the rapidly changing temperature within 3 minutes. First 98, again 50 to 65 and 72. 98, 50 to 65, 72. Like this, a rapid changing of temperature takes place within 3 minutes. Each cycle span for 3 minutes, and within these 3 minutes, there is a changing of temperature takes place. 3 times changing of temperature, and the lid temperature should be 105 degrees centigrade, because the running PCR cycle, the temperatures for PCR cycle is should be less than lid temperature, because if the lid temperature is less than the PCR running temperatures of PCR, the temperature may escape from outside, from reaction to out. So in order to prevent the escaping of temperatures or maintaining the constant temperatures in the PCR reaction, we put the lid temperature at 105 degrees, which is greater than the normal running PCR running temperature. So always lid temperature by default we should be putting 105 degrees centigrade. And why the rapid temperature changes occurs in the PCR, Meso-Peltier effect. That is known as Meso-Peltier effect. This is the physical phenomena which is used in the PCR in order to rapid changing of the temperature. That is Meso-Peltier effect. And the person who is Gary Mullis, who is the inventor of the PCR. He is a biochemist. He is the Gary Mullis, inventor of the PCR. And these are the different types of PCR, so different uses. There are so many number of PCR. There are almost more than 100 patents in the PCR, on PCR. And there are different types of PCR which are used for different purposes. And most commonly used one is RT-PCR. that is reverse transcription PCR, that is used for expressional study. Expressional studies means which type of cells producing which type of expressing which type of genes or which type of RNA. We can identify using this reverse transcription PCR. This is also known as semi-quantitative PCR. In this case, we want to find cell or tissue expressing particular gene or not. We can run RT-PCR and we can find. For example, how we can know only liver cell producing detoxification related enzymes, or beta cells of the pancreas producing insulin, or alpha cells of the pancreas producing glucagon. How we can came to know how specific type of cells producing specific type of proteins or RNAs. By using RT-PCR technique, we can determine this. If you take one cell, skin cell, and another one is beta cell of the pancreas, we can get amplification for the insulin only in the beta cells of the pancreas, but not skin cell. We came to know that only beta cells of the pancreas can produce insulin, and the skin cell is not producing. How can we know? Using RT-PCR, we can using that point. And applications of PCR. There are several applications in the using PCR, and both basic research and applied research. Basic research is the research which is used for the finding of the living process. What are the actual biochemical process or biological process actually going in the living organisms. That only we can know. That is only basic research. Whereas applied research is rely on the basic research. Applied research produce some goods or equipments or things for betterment of the life. So basic research, applied research is relies on the basic research. So both basic research and applied research, there are several applications of PCR. In mutation screening, we can use PCR. Drug discovery, we can use PCR. Classification of organisms, that is taxonomy, we can use PCR. Genotyping, molecular archaeology, molecular ecology, bioinformatics, genome cloning, site-directed mutagenesis, gene expression, all these things we can use PCR. And applied research, genetic matching, detection of pathogenesis, prenatal diagnosis, DNA fingerprinting, gene therapy, all these things we need to use PCR. Recently, real-time PCR used for the COVID testing, COVID pandemic situation. In order to identify in most accurate manner, we use real-time PCR for the identification of COVID-19. In this COVID-19, how we can perform this test means, first we obtain nasal sample from the patient and take into the vial. From the vial, you need to centrifuge first. After centrifugation, whatever the virus in the sample become precipitated, and from that precipitate we need to isolate RNA. From that RNA we run real-time reverse transcription PCR that converted into cDNA, and that cDNA, by adding specific primers, that is virus specific primers, by adding virus specific primers and certain types of fluorescent dyes, we need to run the real-time PCR. During PCR running, if the green one is the test sample and the blue one is virus sample, if the blue line is present, come along with the green line, then the virus is present. If the purple line is not come, blue line is not come along with the green line, there is no virus. Like this, we can identify the presence or infection of COVID-19 using RT-PCR technique. First, we take the nasal sample, centrifuge that sample, and precipitate the virus, and from this precipitation we first isolate RNA. From that RNA, we convert it into cDNA, and the cDNA used for amplification purpose by adding specific primers along with fluorescent tags. While running RT-PCR, it will give whether a virus is present or not. We can easily identify. This is most commonly used method for the COVID-19 testing. There is a real-time PCR. This is a great application of PCR in nowadays. There are several applications of PCR. Thank you. This is all about PCR and its types and applications.